terrestrial leaf litter plays a very important role in the ecology of streams and freshwater bodies. The way it plays a role is that it's actually the main organic matter, which is the leaf litter, when it flows into a pond, forms the food source for a lot of the living things in the pond or a stream system. Now what is happening is, over time, because we are cutting down trees, we are removing grasslands, these kind of forests have typically been replaced by monocultures. So now what happens, because the monoculture is, a lot of the leaf litter which comes in is only of one single species. And to make it even worse, many of these species which are planted around us, for example eucalyptus, acacia, they are non-native. So eucalyptus, acacia, these are the typical plants you will see planted around in Bangalore. And being non-native, when these leaf litters actually flow into the water bodies, many of the organisms which are there have not really evolved with these plant species. So the aim of my study was to actually look at how different kinds of leaf litter affects metamorphosis, growth of amphibian, particularly looking at a burrowing frog called Microhyla ornata, and also how it affects the population dynamics of zooplanktons such as Daphnia, copepods, ostracods. So we conducted a Mesocosm experiment in the field. So what essentially we did is we set up 18 tubs which are each 30 liters in size and sets of 3. One of the tubs in a set had 8 grams of eucalyptus leaf litter, another tub had 8 grams of acacia leaf litter, which is Acacia auriculaformis, which is from not native to India, and the third had no leaf litter, which was acted as a control. In addition, what we also did is we added around 0.6 grams of cat food. So this is just to add some of the organic matter so that the algae growth actually happens faster. And then we added 25 liters of water in each of these tubs and 7 tadpoles within each of these tubs. In addition, what we did is, because we wanted to look at the effect of zooplanktons, we added 1 liter of pond water into each of these tubs, and within this pond water there was ostracods, copepods, Daphnia, all the other zooplanktons what we are looking at. So in terms of sampling and monitoring, what we did is every other day we would go out and look at the survivorship of these tadpoles in each of the tub over a 3-month period. Also every week we would actually count, do a measure of relative abundance of these zooplanktons in these tubs. And also we looked at the water quality, so measurements such as pH, your dissolved oxygen, conductivity over every week for 3 months. One was very clearly that there was no difference in the survivorship of the frogs or the time the frogs took to metamorphose between these 3 treatments. So in eucalyptus, acacia as well as control, they took the same amount of time to metamorphose, and the body weight at which they metamorphosed was also the same. They survived in the same manner. But what we did find is, for the zooplanktons, there was significant differences between the treatments. So while all the 3 had a dynamics over a 3-month period, copepods, ostracods and Daphnias, population fluctuated over time in the 3-month period. But what was key is that while ostracod and copepod numbers was not affected by these leaf litter treatment, but Daphnia, which is an important food source for many aquatic organisms, their numbers reduced in treatments with acacia and eucalyptus leaf litter compared to our control. We also found that the leaf litter treatment also significantly affected pH, water pH level. So one key important thing to remember is, so even though we did not find a significant difference between things, there are very few empirical studies in India which have actually tested the effect of non-native leaf litter on reproduction and metamorphosis of amphibians. And it is crucial to think about amphibians because globally there is a decline in amphibian numbers. So there is a huge push to understand what are the factors which would potentially affect amphibian population dynamics. And since temporary water bodies like the kind of mesocosms we have designed are one of the main sources for reproduction of amphibians and such as frogs and toads. Understanding the dynamics within these systems and the effect of both native as well as non-native leaf litter in these systems is an important field of research. Particularly very relevant to India because India does host a lot of high diversity of amphibians globally. So the amount of endemism in India of amphibians are quite high. So it's very important to understand what are the actual factors which affect their breeding biology.